All right, this is Joey Savage, and on this episode of Supplement Lab, we're finally going to get to talk about Cheat. So, this is going to involve the insulin signaling pathway, AMPK, and a couple other fun things we're going to talk about on this episode of the Supplement Lab. These pathways are really complex. I am, by all intents and purposes, omitting the details that are not going to be pertinent to the ingredients that are in here and what we're actually going to discuss on this. So this is more complex. I'm simplifying the heck out of it. I'm going to tell you about a whole bunch of different 3- and 4-letter acronyms. And yeah, so getting right into it, this is the insulin signaling pathway. Insulin on its own is a peptide hormone. It's responsible for glucose uptake and the downstream effects that occur past the insulin receptor. Now, the insulin receptor is actually made out of two different types of subunits, the alpha subunits that are mounted in the plasma membrane, and then the beta subunits, which are located just below that. When insulin comes in contact with this, it causes a 3D conformational change, and this beautiful phenomenon occurs at the beta parts of the receptor called autophosphorylation, which means that they don't necessarily need to be catalyzed by anything in order to phosphorylate themselves and transmit that signal further down the pathway. If you think about this, it's like dominoes that you've lined up and you're going to knock them all down, right? Well, usually insulin is usually that first domino that gets knocked over and to the rest of them. However, if you skip that part, you can actually knock down the rest of the dominoes while omitting the first couple of steps, which we'll get into. So, after the 3D conformational change in the alpha subunits, the autophosphorylation in the beta subunits, you have this third thing called the IRS. There's 4 different IRSs, which are called the insulin receptor substrates. Now, they regulate a few different things, and activation of the insulin receptor substrates can knock all the rest of those dominoes as well. This is usually a protein that's going to be inhibited by something called PTP1B, which is a protein tyrosine phosphatase. By blocking that, you also allow the rest of the dominoes to flow. So going from the insulin receptor substrate, it activates something called phosphatidylinositol-3 kinase. It activates PIP3 and PDK, which is pyruvate dehydrogenase. These two things will activate protein kinase C, but after that, it'll activate AKT, which PKC and AKT will also activate AS160, which is the trigger event that actually gets the GLUT4 glucose transporter proteins to go ahead and go up and embed into the plasma membrane. After AKT and after this whole glucose transport thing, it has effects on glycogen storage. It will inhibit glycogen synthase kinase 3B, which is something that normally represses glycogen synthase. So if you inhibit something that inhibits something else, you allow glycogen synthase, for example, to make more glycogen. Now, there's this other thing called glycogen phosphorylase. It's responsible for the conversion of glycogen back into glucose, a process also known as gluconeogenesis. It's also a natural process that occurs whenever your body actually needs to generate more glucose as fuel when you have plentiful amounts of stored glycogen. Second pathway we're going to talk about is AMPK. Now, AMPK stands for adenosine monophosphate, or AMP, activated kinase. So usually when cellular energy levels are low, ATP levels are going to be low and adenosine monophosphate levels are actually going to be high. And this triggers AMPK. Now, AMPK is also involved in a lot of energy sensing type mechanisms, especially in the sense of ATP, but it also has a lot of downstream effects. Now, AMPK is something that will inhibit mTOR. So if you're actually looking to increase protein synthesis and get some pretty good muscle gains, an AMPK activation might not actually be something that's viable on your menu. However, if you're looking to increase more glucose and increase glycogen storage, which is an energy kind of thing, then AMPK is a really good way to go in that sense. But AMPK also inhibits cholesterol synthesis. It also inhibits fatty acid synthesis. And it also activates glycolysis, which is the natural process of breaking down sugars to form ATP in the first place. So another thing that AMPK will do is it will block another regulatory protein called TBC1D1. This is something that is another inhibitor that usually blocks the activity of AS160. And like we just talked about over here, when you block something that usually blocks something, you get a net positive. So by blocking this through AMPK, it releases the chains of being held still to AS160. And as we said earlier, AS160 is the actual trigger event that will bring glucose transporters to the plasma membrane so you can absorb glucose. So The third different pathway that we're going to talk about will actually be basically the simple breakdown of carbohydrates into sugars, and this will usually occur at the intestinal brush border. There are different enzymes called alpha-glucosidase, alpha-amylase, maltase, and isomaltase. There's a lot of different enzymes that will break down more complex carbohydrates into actual sugar molecules themselves. The last pathway we're going to talk about, which unfortunately I don't have a drawing for, are the PPAR pathways. So there's four different PPARs. They stand for peroxisome proliferator activated receptor. These are usually different types of receptor proteins that are bound to your actual DNA, and they control the expression of different genes involved in adipocyte differentiation, activation of beta oxidation, and uncoupling, so you can even override the whole beta oxidation thing. But PPAR gamma is going to be referred to, that's the one that is for fat cell differentiation. You want to make more fat cells, activate PPAR gamma. The second one is PPAR alpha, which is a huge range of different effects. However, for our intents and purposes, we'll talk about it in the sense of fat oxidation genes, or the genes that are necessary for beta oxidation. The last one, which is probably my favorite, is PPAR delta, which is also involved in beta oxidation, but it's also involved in uncoupling proteins, which are the override of actual glycolysis or beta oxidation, and you have the spontaneous generation of heat. This is also something that's going to be activated by exercise. A lot of PPAR delta agonists are also referred to as exercise mimetics because of this. Now that I've bored you with 4 different pathways, we're going to go ingredient by ingredient, stuff you're familiar with to stuff you're not so familiar with, and it will always be in the context of insulin signaling pathway, AMPK pathway. Is it playing with the sugar metabolism at the intestinal brush border, or is it a PPAR activator, deactivator kind of thing? So, chromium. Everybody knows chromium. Chromium's probably in just about every GDA that exists. In ours, we have chromium dinacosystinate, which, according to the literature, has been proven to be the most efficacious form of chromium, and they pretty much attribute that to the actual extra cysteine that's there with the xychrome. Now, chromium is something that will activate AMPK. Theoretically, in all the other research that was done previously, there was a lot of thought that went into why is chromium something that increases glucose sensitivity. It actually increases the translocation of GLUT4 to the plasma membrane, but it doesn't embed. Now, AMPK can actually cause the embedding of GLUT4 into the plasma membrane, but it's really something that's going to be dictated by insulin. So chromium, in that sense, will have a synergistic activity with insulin if you have normal insulin levels. So not only does it do that, it helps the GLUT4 translocation, but it also works on this protein here called LKB1. This is something that is sensitive to the AMP to ATP ratio, but is also an activator of AMPK as well. So the net effect of taking chromium is it decreases membrane cholesterol. And by decreasing membrane cholesterol, you actually have a more fluid plasma membrane where the glucose transporters can more readily embed into the cell membrane itself. And that's basically the mechanism of chromium. It works off the AMPK pathway to increase GLUT4 translocation to the plasma membrane. Second thing that you'll see in a lot of GDA products is cinnamon. Now, to differentiate, there's more than one kind of cinnamon. There's regular Chinese cinnamon, Cinnamomum cassia, and then there's the Ceylon cinnamon, the original true cinnamon that's native to Sri Lanka. Now, cinnamon is something that has been shown to increase expression of the insulin receptor. It's also been shown to increase the expression of the insulin receptor substrate. In addition to that, it increases hepatic glycogen, but it usually does this by increasing glycogen synthase expression, this guy here, and it also will be something that will inhibit GSK3B. So by inhibiting the inhibitor and by also increasing the expression of the glycogen synthase, you get higher glycogen amounts. Now, it also will activate AMPK. Now, there's a lot of different active ingredients in cinnamon, and a lot of people it's a controversial topic as far as what actual active ingredient in cinnamon bark actually does the work. And it's not a real clear answer because it appears to be more than one active component doing it. But cinnamaldehyde, a main component that's in cinnamon bark, is something that will activate AMPK and cause GLUT4 translocation once again to the plasma membrane so glucose can be brought in. Third thing is berberine. Now, a lot of people like berberine. Berberine's super hot right now, not only because it's an AMPK activator, but because it has effects that are comparable to metformin. There have been studies for a lot of these ingredients that show comparability to metformin, but specifically berberine is the one that's gotten the most fame and hype. Now, it will increase the actual genetic expression of two different subtypes of AMPK, both the alpha form, which is in the cytosol, and then the beta form, which is inside of the nucleus. It will also increase GLUT4 translocation, like many of the other ones, through doing the same kind of pathway, but it also increases a smaller subtype of glucose transporter called GLUT1. It also increases insulin receptor expression, it increases IRS2 expression, and it's also a PPAR alpha and delta, not an agonist, but something that also increases the expression of those two genes. And as I had said earlier, those are for beta oxidation, uncoupling, and more of the finer genes that control that. But it's also an inhibitor of PPAR gamma, which is the fat cell differentiation gene. So it shuts off the ability to do that to varying degrees. Next is maslinic acid. Maslinic acid is something that increases insulin receptor beta substrate, or insulin receptor beta autophosphorylation, which is that second domino in the line knocking down the rest. And by virtue of that, it will increase GLUT4 translocation. It also will activate AKT through that same mechanism. It will also, by doing that, inhibit GSK3B. And it will also inhibit the two different enzymes in the stomach, alpha amylase and glucosidase, that are responsible for breaking down higher order carbohydrates into their single subunits. Cortisol Ik acid, which is the next one on this list, is something that inhibits PTP1B. And as I said earlier, PTP1B is something that inhibits the autophosphorylation event of the insulin receptor beta. So by blocking a blocker, once again, we have a net positive effect, which allows the autophosphorylation of insulin receptor beta to occur. There's also the ability for this thing, because of this autophosphorylation event, to activate the downstream stuff here. Obviously, once again, we have glucose transporters being delivered to the plasma membrane. And it also works over here on LKB1, just like the Zychrome we talked about earlier. But if this is something that's going to activate AMPK, we have a completely separate mechanism where GLUT4, once again, is translocated to the plasma membrane. It also inhibits alpha-glucosidase, like I just said about maslinic acid at the intestinal brush border. And it is also an inhibitor of PPAR gamma like berberine is, so it stops fat cell differentiation to a varying degree. Last ingredient we're going to talk about is bitter melon, which is probably my favorite. It also will inhibit PTP1B, allowing autophosphorylation and insulin receptor beta subtype. It also increases IRS1 phosphorylation right here. And because of that, once again, we're knocking down the rest of the dominoes in the line. We also have GLUT4 translocation as a downstream effect. But something that's unique about bitter melon is it works off of this adiponectin thing, which is what is called an adipokine. It's a signaling molecule that comes from fat cells. But adiponectin is something that will cause a downstream cascade where it activates AMPK. Once again, this is going to be inhibiting cholesterol synthesis, triglyceride synthesis, and so on and so forth. But in addition to that, we also have the activation of PPAR alpha and gamma. Now, the weird thing about bitter melon, when you're activating PPAR alpha and you're increasing beta oxidation and also activating PPAR gamma, you have more fat being burned, but you have an increase in cell number. And the clinical outcome of this is you have an increase in total number of fat cells, but they're smaller. They're much, much smaller. So it's a weird kind of trade-off with bitter melon. In addition to that, the downstream genes that are affected by PPAR gamma activation from bitter melon, like, what is this? Like fatty acid synthase and acetyl-CoA carboxylase, those downstream genes are actually downregulated. So even through some weird kind of finagling of the PPAR gamma gene, we can end up getting activation, which ends up making more cells, but a compromised ability for them to actually make fat. So that was kind of weird, but it's cool. The last thing about it is it's a PPAR delta activator. Now, the PPAR delta is probably the most special of all the different PPARs that exist. Bitter melon had an effect on PPAR delta, and it increased its expression about 1.5 times over normal levels. In addition to that, it also increased the promoter region protein that actually helps the transcription of PPAR delta at 1.5 times as well. So once again, PPAR delta is something that's going to increase fat oxidation, but it's also something that's going to increase uncoupling protein, which is something that's going to end up increasing the overall energy level and thermogenic effect of this. Now, on taking this product, now with Cheat, it's a 6 capsule a day dose. We recommend that you probably, if you're going to do 3 meals a day, do 2 capsules 1 hour prior to consuming food. And we say this because it's in a DR cap. Now, once again, it takes about 60 minutes in order to get to the small intestine. So you want to take these things at least an hour before consuming food. If you're not eating 3 meals a day and you're probably doing 2 and some snacks, you'll want to take 3 capsules before your largest 2 meals of the day. But other than that, you know, this is going to cause a noticeable, you know, decrease in blood sugar. You will probably get a little hypoglycemic. So you don't want to take this stuff and not eat, because you're probably going to end up wanting to take a long nap. It just will naturally make you tired because your energy levels get so low. But this could shift you into more of a fat burning state. But at the same time, if you do consume carbohydrates, this whole thing is more conducive to making you store more glycogen and burn more fat than not doing anything at all. And one last thing about this stuff, Berberine, this is just because I like doing cool things, is something that naturally fluoresces under black light. I don't know if the camera can see this. Can the camera see this? Okay, so yeah, these things will glow if you put them under a black light. It's just a natural phenomenon, nothing crazy. We didn't have to really innovate really hard. We just had to use berberine in order to make this happen. Anyway, so if you want better glucose absorption, we're doing it in like 6 different ways. The other 2 things that are in this product are pine bark anthocyanidins, which are really good for blood flow, and this other thing which has pomegranate in it. And if you know anything about galactanids and ellagic acid and what happens when it comes into contact with your microbial colonies that live inside your small intestine, that'll be a little breadcrumb for you that we'll probably talk about in a later video. But if you do a little digging on this, you might find something cool. Anyway, that's been Cheat. Enjoy this. Wait an hour, then go eat. We'll see you next time on the Supplement Lab.